Okay, thank you once again, everyone, for joining us in today's webinar. This is our first webinar in this series of molecular biology and immunology techniques. And we are happy to have with us our colleagues who is a PhD student at the Casas Medical Center, Alfred, joining us to set the ball rolling for us. And if you are joining us today, it's brought to you by the Elliott Research and Data Science Institute, ERDSI or XC, and you are always welcome to join any of our upcoming webinars, so far as it's free or affordable. Okay, Alfred, over to you, and
All
we
right,
hope
thank
you
you,
all
Eben.
enjoy today's session.
Yeah. All right, thank you. Let me
Mhm.
share my screen. I think I can share the entire screen. Yeah. And please let me know if you can see my screen.
Yes.
Okay. And do I need this? I'm not sure I need this one. No problem. Let me see if it doesn't disturb. All right, Eben. So thank you for this opportunity and also the platform to learn and also have discussions on such techniques. So one thing I've seen over here is there are a lot of techniques that, in fact, those of us from Ghana, should I say Africa, we don't know much about. We have no idea that these techniques do exist. And so I was pleased when you mentioned that we should have this kind of platform to share our knowledge regarding some of these techniques. So I'm sure some of you too are interested in coming to graduate school and you do write SOPs. Sometimes you do get rejections. One thing I saw during my time of application was you being able to figure out what your future research direction will be. So assuming that you did a particular project at the undergraduate level, and then you've taken the time to write about it in your SOP. Now it's up to you to also include or add in the SOP what your future direction is going to be. I mean, in case of the research. So if you're able to see the future direction and the techniques you're going to use to, you know, achieve that kind of results, they do really appreciate it. And so I thought that it's good or wise enough for us to okay or conversant with some of these techniques in case you want to apply for schools. Those of us already in schools too, sometimes you perform a particular research or experiment and you are asked to do something else. Okay, so if there's the need for you to employ any of these techniques, of course, you would have some sort of basics. So that's why we decided to have this series. And today, one of them is co-immunoprecipitation. Um, that's the main technique for today. And after that, I will be sharing or having a paper discussion with you. I'm trying to share my, I'm trying to use a pointer, but I don't know. Uh-huh. All right. Eben, can you see my screen? Like, can everyone see my screen? Is it working?
Yes,
Okay.
should be
And the
good.
pointer too.
Yes.
That's great. So I will be talking about co-immunoprecipitation, what co-immunoprecipitation is, what we use it for, and how are we going to use it to answer a particular research question. So that's it. Then I'll be using this paper as an example. A paper that was published in 2021. It's used co-immunoprecipitation a lot. In all of their figures, you could see that there are a lot of co-immunoprecipitation techniques in there. So yeah, without wasting much time, I think we can begin. So I'll talk about the types, the workflow for immunoprecipitation, sample types and then preparation, antibody selection, which is very important. And we talk about how you're going to analyze the result and then look at how to interpret your results. And we consider the paper discussion. Now you can jot down your your questions at the end, then you might feel free to ask me. You can also drop them in the chat if you don't want to forget about it. Then you can put it in the chat as well. So what then is immunoprecipitation? When we say immunoprecipitation, basically it's an antigen antibody reaction. So there's an antibody, there's an antigen, and then you want to detect this antigen. Of course, there's the need for you to develop an antibody against this antigen. Okay, so immunoprecipitation is basically an antigen antibody reaction, whereby we use an antibody to detect the presence of something. So in a cell or in a tissue, let's say you have breast tissue, you have colorectal, tissue or colorectal cancer tissue, you have a cell lysate, and you suspect that there is a particular protein or antigen in it. You just have to develop an antibody against it, and you're able to detect that particular antigen of interest. So basically, that's all about immunoprecipitation. So we use it to investigate the presence of something. Like I mentioned, if you suspect there is, so when you take the ABO blood group system, we can detect our blood type by using antibodies against the antigens that we have in our system. So it helps us to detect the presence of something. The relative abundance. So sometimes to after performing this immunoprecipitation, you can go ahead and then look at how the quantity that is in there by using ELISA and other techniques as well. Now, when we also want to know the size, if you are interested in a particular protein and then you want to determine the exact molecular weights, you can use immunoprecipitation. Now, up regulation and then down regulation. So assuming that you have a drug that you think might have an effect on a particular the expression of, let's say, protein A. So prior to administering this drug, you measure the level of the protein. Then after giving the drug, you also measure. So it's kind of before and after. So it's a protein. You are detecting the level before the drug and then after giving the drug. Now we'll be able to tell whether there was an increase in the expression or there was a decrease. That's what we talk about, refer to as up regulation or down regulation. Stability. So sometimes you want to look at how long a particular molecule stays around in our system or under various conditions. So if you want to know how long it sticks around before degrade, we can use immunoprecipitation. You can also look at post-translational modification. So after translation, you know, a lot of molecules are being added to proteins. Now, if you want to know whether there was a phosphate group somewhere that was added, whether there was a methyl group that was added, you can use immunoprecipitation for that. And what you are interested in today is interactions between proteins. Now, you suspect that protein A is interacting with protein X. How do we determine that? We can use immunoprecipitation to look at the interaction between two different proteins. Okay. So what are some of the types of immunoprecipitation? We can have individual protein immunoprecipitation. Individual in the sense that we are interested in just protein A. So we have protein A here. We want to find out whether it is present or not. So all we have to do is to develop an antibody against protein A. And if it's there, we will see a band. If it's not around, when we perform, let's say, electrophoresis, we will not see any band. So once it's present, the antibody will bind to this protein and we are going to see it. We can visualize or we can even quantify. That is just one protein. Now, the co-immunoprecipitation, which is complex protein immunoprecipitation, here we want to find out if this protein is interacting with this. So this is our protein A and this is protein X. And we believe that this protein is interacting with the one we have here. Now, what we can do is we can develop an antibody against this protein. Now, after the washing steps and everything, if there was an interaction, we can stain and then see the presence of this protein X. We can see a band for this protein X. So that is all about co-immunoprecipitation. We want to find out if proteins are interacting. We develop an antibody against this protein, and if it was interacting with this, we should see this when we also use a different antibody for this. We can do it the other way around. We can develop antibodies against this, and then we expect to see this at the end of our experiment. Now, other types of immunoprecipitation is to look at what proteins are being bound to, let's say, the DNA. So we have chromatin immunoprecipitation. So we have the histone. Histones can be modified by methyl groups and other things, acetylation and what have you. So we want to find out if there is anything that is bound to our histone. We can perform CHIP assay, which is chromatin immunoprecipitation. So here, this is our DNA. Is protein A binding to it? What we do is that we cross-link, we fix to make sure that yes, it is bound, it is binding to it. And one, it is binding, we just have to develop an antibody against this protein. And then in the end, we are able to tell that yes, when we take this sequence, this protein was binding at this site. What if the protein is also an RNA binding protein? So if it is binding to a particular RNA, we can do RNA immunoprecipitation, which is this one. And so that is basically all about immunoprecipitation. And there's one that we call IP with tagged proteins. So we can, instead of developing antibodies against, let's say, protein A, if we don't have an antibody that is strong enough to recognize protein A, we can fuse protein A with a different protein, then develop antibody against that particular protein. So we tag protein A with something else, then we call it by that thing. So that's what is meant by tagged proteins. All right. So the individual, so let me hide this. I think I don't need this. Okay. So individual protein immunoprecipitation. I'm just going to walk you through the steps or the workflow. It is very simple. So here we are interested in just protein A. This is our protein A. Let's say this is protein B or other substances in the cell lysate. So we take a sample from somebody. It can be blood, it can be cell culture, it can be a tissue, it can be anything. So you take the sample and then you suspect that protein A is present. Now what we do is we find an antibody that specifically binds to protein A. So that's what we can do. That is one way. We just incubate our antibody with our cell lysate. Now, another way we can do this is also by using antibodies. We add beads to the antibodies, okay? Now, in a minute, I'll tell you the purpose of the beads. So, one of two ways. First of all, we can incubate antibodies with our lysate. Then we add the bead to it. Now, the bead will recognize the FC region of our antibody. Now, what we aim to get in the end is this complex. So that is our antibody, which is bound to protein A, and then we have a bead here, so that when we wash, we don't lose our antibody and then the protein. Now, the first portion was to incubate your antibody directly with the cell lysate. It binds to the protein of interest. We add a bead, which also binds to the FC receptor of our antibody. Now, the second step is we already incubated the antibody with our beads. We add the sample, and then we still get the same. So you can decide to go by this way or use this way. In the end, the aim is to form this complex, whereby you have an antibody which is bound to your protein of interest with a bead. Now, what we do next is to wash. Now, you know, we have this complex. Our interest is to look at this protein, the presence of this protein, not this or that. And so we wash to get rid of the debris. So all the other things, even the lysis buffer we used over here and everything, the detergent we had, we have to wash all of them away. So after the washing, we expect to see something like this. Only this complex, your antibody, the protein of interest, and then your beads. Now, when we have this, the next thing to do is to get rid of the beads, okay? The beads and then the antibody. So we use a different reagent that has higher affinity for our protein of interest. So we elute the antigen or the protein that we are interested in, let's say protein A. And so we believe that after washing all the debris away, washing all the other proteins that we were not interested in, we should find only the protein of interest. That is if it is present. And here that is protein A, and then it's in this tube. So let's call this a tube. We have our protein of interest here. How do we analyze? We can perform a Western blot whereby we use antibodies to stain or to react with this so that we can visualize it as a band. Now, one thing you should take notice, when you want to analyze, you should have an idea of how or the size of your protein of interest. So there's a ladder here. Now you can tell if your, let's say your protein has a molecular weight around 120 something, and you think this is 120 something, it corresponds to your ladder, then we know that this is your protein of interest, and we are going to see a band here. That is when we perform the Western blot. So that's how it's done. This is for one protein when we want to test whether protein A is present or not. So if it's present, we are going to see a band. If it's not present, in the end, we will not see any band because nothing will be bound to this antibody. And when we wash away, we will have only antibodies. When we perform the Western blot, we are not going to see any protein here. That is when the protein is not present. Now, that is that for just immunoprecipitation. That is when you are interested in just a single protein. Now, what about when we have two proteins that are interacting? We want to find out. We believe that, let's say, in a particular tumor tissue, we believe that protein A is interacting with protein X, which is making the diseases more, having the ability to infect, or let's say, metastasize into other tissues or move into other tissues. That's your hypothesis. You believe that breast cancer tumors, they do have these two proteins that are interacting, and that's why we are seeing the aggressiveness of this particular condition. How do you confirm it? How do you tell us? How do you convince us that indeed these proteins are interacting? That is when we use the co-immunoprecipitation. So it is used to analyze protein-protein interaction. Protein A, let's say protein B, protein A to, let's say, protein X, and what have you. So here, the main purpose is to identify interaction partners. So we are interested in protein A, but we also want to find out if protein A is interacting with protein X. So it can be any other protein, it can be a cofactor, it can be any signaling molecule that we think the protein is interacting with. Now, when you take transcription, for example, we have the RNA polymerase, and a lot of things come together for transcription to occur. Now, you might hypothesize that, oh, you believe you found a new protein, let's say protein X, that kind of interact with the RNA polymerase. That's why maybe it's halting transcription or not. So in order to confirm this, then you will need a co-IP to confirm this interaction. So take note that Co-IP is to use, it's used to look for interaction between two proteins. Now, it can be used on cell lysate. It can also be used on tissues. So we can take tissues and then we homogenize the tissue. We stain with antibodies to know whether proteins are interacting or not. We can also have a conditioned media and we can perform co-IP on it. So now let me take you through the workflow, which is similar to that of the individual immunoprecipitation. So now this is still our protein A, but we suspect that the yellow one is, let's say, protein X. We believe protein A is interacting with protein X, okay? And so we develop antibodies against protein A, only protein A. In other way, we can also develop antibody against protein X. And if they are interacting, we should see this partner whereby we are seeing protein A being bound to the protein X as well as the antibody. So note that this one too can be done in one of two ways. The first one, you incubate your cell lysate with the antibody that detects protein A, you add your beads, you form the complex. Or you can incubate your beads with the antibody, you add the sample, you still form the complex. So now it's not only, it's not just a complex of protein A, antibody, and then the bead. Here we have protein A as well as its interacting partner. Now protein A, we are using protein A as a bait. So it's like you're going for fishing, you are using a particular food as a bait. So if the fish sense that kind of food, they come around and you are able to capture the fish. So that's what we do here. We want to know whether protein A is interacting with B, or let's say protein X. We use this antibody for the capturing. So we use it to capture protein A. And if this protein X is interacting, it should form a complex with the antibody. So that's what we see here. Now, whatever we see in here, these are all debris, and so we are not interested in this. So we have to wash. Now, when we wash, okay, we remove all the debris from it. We are left with only the bead, antibody, protein A, and the interacting partner. So this is our bait, and then this is our prey. So we have the complex here. Like I mentioned earlier on, we also use a buffer which has higher affinity for our protein of interest. And so we elute it from the beads so that it doesn't interfere with our gel or the Western blots that we perform over here. So now we use antibodies against A. And we suspect that if A was interacting with protein X, we should see protein X here. Assuming there was no interaction, then we shouldn't see these yellow things here. And so we are going to use antibodies against X. So over here, not A again, but rather against X. Now, when we are able to see protein X here, then we can say that, yes, there was an interaction between protein A and then X. So take note, you use antibody against A, you wash, you get rid of everything. If there is an interaction, we should end up with protein X here. That is, if there is an interaction between protein A and then X, we should see X here. So when we stain for X, we should see X over here. Now, if we don't see X, it tells you that there was no interaction between protein A and then X. We can also do it the other way around, whereby we use antibodies against protein X. Now, when we get here, we stain for protein A. And if we see A, we can confirm that there was an interaction between protein A and then protein X. So like I mentioned, if you have any question, you can write it down. You can also put it in the chat. I will address all of them after this. Yeah. So one key thing we are also interested in, this is an interaction between two different proteins. And so the main goal is to ensure that the complex is there. The protein-protein complex is there. The interaction is not interrupted. Now, once we distract the interaction between the two proteins that are interacting, okay, we are not going to see the interacting partner here. So we want to make sure that this interaction, the bond between protein A and then X should be maintained throughout our system, okay, so that in the end we can visualize. So that's why the sample types and the preparation is very important. So this can be done on cultured cells. You can culture your own cells, use cell lines, transfect these cell lines with proteins that you are interested in, that you think are interacting, okay? You transfer them with these proteins, and then you perform the Co-IP to see whether the proteins are going to interact or not. You can also use tissue. You can generate knockout mice, knockdown mice, express the proteins in mice, take, let's say, their liver, the spleen, any organ that you think that's where the protein will be expressed. Now you perform Co-IP, and then you see whether there was an interaction or not. So if you are working with cells, of course, you wash you add your lysis buffer to the culture, and then you proceed with the experiment. If you are working with tissues, make sure to homogenize the tissue, because some of these proteins might be bound in a particular membrane. So what is the main important thing is to homogenize the tissue, expose them so that your lysis buffer can actually work on them for you to get access to the proteins of interest. Now, you can, the lysis buffer can be done with agitation. So mostly we have a router whereby you place your sample and everything on it. It just kind of makes it gently for you over time. So it depends on the incubation period, and also depends on your protocol, how long it's supposed to be on it. Now, the lysis buffer is important, and so it depends on the sample type. The type of sample you are using determines the lysis buffer you are supposed to use, and the protein localization. If your protein is in the cytosol, you don't need a very harsh buffer. If it's in the nucleus, of course, you should find a way to permeabilize the various membrane in order to get access to your proteins. Okay, so mostly read about the protein, know the location of expression, know the type of buffers that have been used. Okay, they always say that there is nothing new under the sun. Whatever you want to do, there are protocols. Optimize the protocol, make sure it works well for your experiment. Now, the main goal of whatever we are doing here is to maintain the native protein conformation and then the protein-protein interaction. So, you know, proteins, they have their individual properties. They are made up of their primary structure. We have the amino acids coming together, and with their individual properties, it helps the protein to form the tertiary structure with a particular conformation. So if there is a conformation, maybe in that particular conformation, that's when the interaction occurs. Once you distract this conformation, once you denature the protein, we are going to miss the protein-protein interaction. That's why you are supposed to use a buffer that will maintain the native protein conformation so that the interaction between the two proteins is maintained. We don't lose the interaction. Once we lose it, we are going to end up washing the interacting partner away. In the end, your result will be that there is no interaction between these two proteins. Meanwhile, there was an interaction. Okay. Now, you know, proteins too can be degraded by proteases. So as part of your lysis buffer, make sure to include protease inhibitors, which will prevent degradation of your proteins. Now, the common lysis buffer that I use, we have NP40 Triton X. Like I mentioned, depending on your protocol, look for protocol that has been used and optimize it, make sure it works well for your studies. But these are some key ones. These are mild detergents in the sense that they don't disrupt the native protein conformation, and so the interaction between the two proteins is maintained, and so you can rely on some of these detergents. Now, for the initial step at the lysis stage, you wouldn't want to use SDS or deoxycholate because these ones, they're able to disrupt the interaction. They can denature your protein, so you won't see any interaction between them. So at the lysis stage, we don't use some of these harsh lysis buffers. Now, REPA is one common thing that is used in Western blot. It's good for Western blotting, but not for co-IP, because if you have weak interactions, it will be difficult for you to maintain the interaction. It's a bit stronger, and so we don't recommend using REPA for your co-IP, unless you are sure that the interaction between the two proteins is a bit strong and it can withstand the pressure from REPA. Else you don't have to use it. So washing too is very important. You know, whenever there is an antigen antibody reaction, we also suspect some sort of background noise, and you might want to reduce this background noise that is coming in, which will interfere with your results or your finding. And so washing is very important. You make sure to include protease inhibitors or phosphatase inhibitors, so that the interaction is also maintained, which is very important. Now, one thing is that when your washing steps, or when your washing is too mild, okay, you end up with a very high background, which in the end you might have to come back and then optimize again. And when it is also too harsh, it means that you're going to lose the interaction. When you have very weak interaction, you're going to disrupt. So it should be kind of moderate, in between mild and then harsh. So it all comes down to the point that you have to optimize the washing steps, all the steps, the lysis buffer, the washing steps, even the antibodies you are working with, the incubation period and everything. Make sure you modify, because it is very important. And one thing we can also include as part of the washing buffer is DDT or BME. BME is beta-mercaptoethanol. It usually breaks the disulfide bond between proteins. So, you have a cell lysate, there are a lot of proteins in there, and sometimes they do form this disulfide bond between them. So what you can do is, because we are interested in only protein A and then protein X, we can include DDT or BME. They disrupt this disulfide bonds between the other proteins, because our interest is to get only protein A and then B and or X and nothing else. So we can include these things too. So if you see them in any protocol, DDT or BME, they just disrupt the disulfide bond between proteins. Now, the antibody selection is also very important. Specificity is key, because if you are interested in protein A and you end up detecting protein B, which is known to already be, let's say, an interacting partner of, let's say, protein X, you give us false positive results. So your antibodies has to be very specific, which is crucial. Now, species reactivity, if we develop an antibody against a mouse protein, okay, we cannot use that antibody for a human protein. You are not going to see anything. But when you have, let's say, a mouse and then rat, sometimes they do share, the proteins can share some sort of similarities between them, and so there could be this sort of cross-reactivity. So we make sure that if it is generated against mouse, we use mouse antibody or mouse samples. You don't have to use any other species. Yeah. And then the clonality is very important here. Now, we have polyclonal antibodies, we can have monoclonal antibodies. Now, the polyclonal antibodies, these have the ability to recognize different epitopes of our antigen. So let's say our protein A has different epitopes, let's say 5 epitopes. Polyclonal antibody can recognize all the 5 epitopes, or maybe 3 of them. Now, monoclonal will just recognize just 1 epitope. Now, we want to capture protein A. Protein A is also interacting with protein X. What if the epitope where our antibody is supposed to bind is the part involved in the interaction with protein X? It means that we are going to disrupt the interaction between protein A and then protein X. And so at the capture stage, where you want to detect, so at this stage, where we want to use this antibody to capture this, assuming where the yellow protein is bound to, that is the portion involved in the, that's the portion where our antibody can recognize. Then it tells you that when the antibody comes in, it's going to disrupt the interaction between these two proteins, and we are going to mix their interaction. We will wash and nothing will be bound to this red protein. So in the capture part over here, when we want to capture, when we want to detect, when we want the antibody to bind to protein A, we can use a polyclonal antibody which can recognize a different epitope. So assuming where this epitope is also involved in the interaction, which is involved in the interaction with protein X, then our antibody can find a different epitope and then bound to, without disrupting the interaction between protein A and then X. So that's one thing with antibody selection. Now, in the Western blot, in the end, whereby we want to detect whether X is present or not, we want it to be highly specific. And so over here, it's okay to go with a monoclonal antibody. So best practice, you can use different antibodies for the capture aspect, which is the immunoprecipitation aspect, whereby we develop antibodies against protein A. You can do use polyclonal. Where you want to develop detect protein X, you can use a monoclonal antibody. Now, how do we analyze? Assuming we had everything right, everything is on point, our lysis buffer, washing, everything is fine, and there was an interaction here. How do we see this? How do we confirm that indeed protein X was interacting with protein A? So this is the part where we do the analysis. We can perform SDS page or we can perform a Western blot. So we usually do SDS page. Now we transfer the proteins onto a membrane in the Western blot. Then we stain for protein X. And so I'm going to talk about this portion in a bit. So here, this is our SDS page. SDS page, mostly we use sodium sulfate to denature all our proteins. Over here, we are no more interested in the interaction because we have everything here. All we want is to separate them according to their size. So we can use any buffer at all that can help us to visualize or help the proteins to separate according to their sizes. So here we can use SDS. SDS confer negative charge to all your proteins so that we load them into a gel. So let's say this is sample 1, sample 2, sample 3 in that order. This is our ladder. Now we load everything. Because we use SDS, all the proteins now have a strong negative charge. Now it means that when you subject them to voltage, they are going to migrate from the negative portion to the anode, which is the positive side. Okay, and so because they all have negative charge, they are going to migrate here based on their sizes or according to their sizes. And so the proteins with higher molecular weight will be closer to the starting point. Those with low molecular weight will move at a very high, faster rate to the anode compared to those with higher molecular weight. So that is the SDS page aspect. Now we've separated all of them, all the proteins that we have according to their sizes. Let's assume that this particular size corresponds to whatever we are interested in. So this is protein X. How do we confirm? Because we can have other proteins in the sample that might also have the same or similar molecular weight as protein X. So we must use an antibody that recognizes only protein X so that we can confirm that indeed it is protein X that we are seeing here. So we perform Western blot. In the Western blot, what we do is that we transfer the separated proteins which are on the gel onto a membrane. So it can be a nitrocellulose membrane. We transfer it onto this membrane. Now we put it here. What we also do is we subject it to voltage for like an hour, and then the proteins are going to migrate in this direction. So they were on the gel, they are going to migrate in this direction. They will end up on the membrane. So we will see them on the membrane. So in the end, after running it, you see all your proteins that were here on this membrane. Now we know this is our ladder, and we know the molecular weight of the protein we are interested in. This is protein X. Now we can stain with a primary antibody. We add a secondary one with a horseradish peroxidase. Now we add a substrate. Then we can see a band on the membrane. So we can tell that yes, indeed, protein X is present or not. So basically, that's how the analysis is. After eluting, you have to separate them according to their sizes. Now, you can't just, based on the size, tell us that that is protein X. Other proteins can also have similar molecular weight as protein X. So you should go further and then stain with an antibody which is specific to protein X. Now you can confirm that yes, protein X was interacting with protein A. So what are some of the controls? Controls are very important because we don't want any situation whereby you come and tell us there was an interaction, of which in actual sense there wasn't any interaction. So we include a lot of controls to rule out any false positives. So first one, assuming you are working with cell lines, so you're working with mice, what we can do is that we can knock down protein A. We want to find out if protein A is there or not. Protein A is interacting with protein X. Now, the negative control, one of them is knocking down or knocking out. So we can knock out protein A. And if we knock out protein A, it means that when we get here, if we knock down protein A, it means that when we introduce this antibody, there's no protein A for the antibodies to bind to. And so this complex will not be formed. Nothing will be here, so there shouldn't be protein X here. So when we stain, we shouldn't see protein X because there was no protein A for us to begin with. So it can serve as a negative control when you knock out or knock down protein A. Now we can also use beads only. Now beads without antibody. We introduce beads, no antibody. So like this one, just the beads, there's no antibody. You don't add this antibody. So if there's no antibody, the bead alone shouldn't bind to any protein. If the bead starts binding, then it tells you that there was non-specific binding that was coming from the bead and not the antibody, which rules out that there's no interaction. There was something else that was interacting, which was incorrectly picked by your bead. And so you want to confirm if, so the main goal is to make sure that you had everything on point. Okay, so that is one way we can do. So another negative control is isotype control. You can use a similar IgG antibody, but in this case, it was not developed against protein A. So it's just an antibody, but it wasn't developed against anything. So it's just there. And so if it's not developed against our protein A, then in the end, we shouldn't see X. That is, if there is an interaction, we shouldn't see it because protein A is not around. Now, positive controls, we can use 1 to 10% of our lysate. So when you prepare the lysate, when you lyse your cells, you want to be sure that both protein A and then B, or let's say protein A and X, were present even before any other thing. So you keep some of the lysate, then you perform the Western blot. Over there, because it's just the lysate, it must have both protein A and then X. It doesn't say they are interacting. It just confirms that, yes, the proteins were even there before we even proceed to see whether they are interacting or not. So that can serve as your positive control. So you see input a lot in most of the articles. You see input as a positive control, and in the input we expect to see both the protein and then its interacting partner. We are expecting to see it, unless you decide to have a lysate whereby only the protein A is expressed, and then a different lysate whereby only protein B is expressed. That one too is allowed. You can do so if to set different experimental conditions. So now let's look at how the results look like after performing the Western blot. What do we see when we have a co-immunoprecipitation, when we suspect that two proteins are interacting? So here I'm going to use, let's say the bait here is our protein A. And we want to find out if protein A is interacting with protein 1 and then protein 2. That's the aim. This is our input. It tells you that this input is coming from the lysate. So when we lyse the cells, we just took a portion and then we performed the Western blot. We did not go through all this process. We just took a portion from here we did this. Okay. Now, in this particular result, what they did was that they just transfected cells with either the bait, this protein, or this. So that's what they did. So the first lane, they had cells that they just transfected it with the bait, only the bait. And so if there's a cell whereby we have protein A in it, then when we stain for protein A, we should see protein A. So yes, it confirms that protein A is there, and that is a positive control. Now, in lane 4, that is after everything. So lane 4 is coming from this part, this portion, whereby we are done with everything. Whatever we had here, we just performed the Western blot. So that is the results that we are seeing at the end here. So the whole of this, they performed the Western blot on that side, which is the elute, when they eluted the sample. So now, over here too, they had only the bait, so they decided to, so your cell whereby we had only the bait, it's there. When you stain for it, we should see it. A cell whereby they transfected it with this, when you do the washing, when you do all your steps and everything, we should see that, yes, indeed, the bait is present. So that's what we are seeing here. Now, the next thing is the second lane. What we can see here is that the cells were transfected with just protein 1, protein 2. So in this particular, let's say, sample, we have only protein 1 and then protein 2. So we use antibodies against protein 1 and protein 2, and of course, we are seeing them here. Now, mind you, they did not express protein, the bait, which is protein A here. And so we don't expect to see any band here. And so these ones are serving as our negative controls. Now, the same thing, like I mentioned earlier on. So here, this result, like I said, it's coming from this side. This side. What they did was that they did not express this protein at that portion, at that particular sample. This protein is not present. And so if it's not present, okay, if the bait that we are using it to catch 1 and then 2 is not present, now when we stain, we are not supposed to see anything because protein A is not available to interact with this, and so it's not present. So when we stain for 1 and 2, we shouldn't see them because this is after using antibodies against this, washing everything, and then we try to see whether we will see these proteins interacting with this. Now, we don't even have this protein to begin with. If this protein is not there, it means that when we wash, we will wash protein 1 and 2 away. And so when we stain, we shouldn't see anything here. Now, the other lanes that we have, let's say over here, they express both the bait, protein 1, and then protein 2. They expressed all these proteins. And so if I have a lysate that has protein A, protein 1, protein 2, and when I stain them, I should see all of them. So we are seeing all the bands. Now let's come to this side. Now in this lane, we also had the bait being present. We had the prey, or let's say protein 1 being present. Then we had protein 2 being present. Mind you, we used antibodies against this. And so after washing and performing everything, we will see only the proteins that were interacting with the bait. So here, this is our bait. We used antibodies against it. We are seeing the yellow one interacting with protein A. So let's say this is pre1, it's interacting. Pre2 did not interact. And so here we see only pre1 and then the bait. So that's what we are seeing at the back here. So over here, you could see that we have the bait, we have the prey, and then we have pre2. Now, when we stain for for the bait, of course, it has to be there because it was present. Okay. Now, when we stain for the second protein, yes, we are seeing it, but we are not seeing the third protein, or this one, protein 2. It tells you that protein 2 was not interacting with the bait. That's why we are not seeing any band for it. So we got rid of it during the washing. When we were washing, protein 2 got lost because it was not part of the bait pre-1 complex. It was not part. And so when we stain for it, we will not see it. So from this result, we can say that only protein 1 was interacting with the bait. So that's what we see here. So a lot of controls. We just added all these controls to be sure that whatever we are seeing is the true results and not any false positive or false negative result. So now another example here, let's say we take a sample like mice. We take a tissue from mice. You can take any tissue at all, prostate tissue, breast tissue, and then you have your inputs. What will you use as a control? So we have wild type. Wild type is the normal phenotype. Nothing has been changed. That is the usual thing that we have. So when you took the tissue, you didn't perform any alteration. That is the tissue. So we took breast tissue. All the normal proteins in breast, they should be there. And so we stain for our bait, which we think is interacting with 1 and 2. So we stain for it to see if it is present or not. And because nothing has been done to it, we should see the bait, we should see the antibody protein 1, we should also see protein 2. Now, another control that we can add, a negative control, is the knockout control. Knockout in the sense that we are going to delete the bait. So we take the sample, we can use CRISPR-Cas9 system, we can also use the 3-LOXP system. You can also, a lot of systems, a lot of ways to delete. You can also knock down protein, this bit, which is, let's say, protein A. We will knock it down. This is just the input. This is just the lysate. So let's say from the first breast tissue, we just have the breast tissue as it is. From the second, or let's say from the second one, we knock out this one. Now we perform this Western blot. We see all these proteins present. Now in the knockout, we've deleted this protein, so we shouldn't see any band. So there's nothing there. Now we perform our Western blot. We did our co-IP. We did everything. So this result too refers to this aspect, this sample. So not here. Now we are not here anymore. This is where we are now. We performed everything. We did our co-IP. And then the wild type, we are seeing the bait or protein A in it. We are also seeing protein 1 here. It tells you that in the wild type, whereby we still have our bait intact because we developed antibodies against this, we are seeing a band for this. It tells you that there was an interaction between the bait and then protein 1. That's why we are seeing a band for this. Now, because there was no interaction with protein 2, we don't see any band. So it means that we got rid of protein 2 during the washing step because it wasn't part of the complex between this and then this as well. So now, how do we confirm that it was not interacting with, or it didn't end up there by chance? In the knockout, we don't expect to see any band for protein 1 because we only see it when the bait is present. Now, if the bait is not present, it tells you that we will wash everything away. And so when we stain for you, we shouldn't see you. So this confirms that this is a negative control, confirms that indeed there was an interaction between the bait and then protein 1 and nothing else. So that is how it's done. So when you have any proteins that are endogenously expressed in a particular tissue, you can perform some of these experiments to see whether some proteins were interacting or not. In the controls, you can just knock out some of them. You can also do knockdown, whereby you use siRNA to delete their RNA so that they don't form that particular protein. So that is it. So if the protein is not there, then we shouldn't see any interaction. We only see the interaction when it is there. That is, even if there was an interaction, that's only the time you will see. So here, the bait is present, but it's not interacting with protein 2. It tells you that it's interacting with only this one. So another one is this is our input. Let's say our lysate. So we have our lysate. Of course, all the proteins have to be present. So we have the lysate, all our proteins are there. When we stain, we see them. We use a negative control, which is, this can be an IgG, an antibody that does not recognize the bait. So it does not recognize protein A. So if it does not recognize protein A, then we shouldn't see anything here, because in the washing step, everything will be washed away. So we shouldn't see any band in this lane. Now, after performing the Western blots, we could see that the bait is present. We use antibodies against the bait, it's present, and we can now confirm that it's interacting with only protein 1 and not protein 2. So that's how the whole thing is. Now, someone would want to find out why we have GAPDH. So GAPDH is one of the enzymes in the glycolytic pathway, and because most cells utilize glucose in their activities, it means that this enzyme is highly expressed. And so we use it as kind of internal control to confirm that the whole thing, the chemicals, everything that we are using indeed work. So here we stain for GAPDH. You can also stain for beta actin. It is highly expressed under, all conditions. You still see beta actin. So you stain for those things to confirm that your steps and everything worked. And if there is any negative or any positive here, it is a true thing and not due to chance. Yeah. So now, what are some of the limitations? We will be able to tell that these two proteins are interacting. But what if the interaction is just for a short period? So when you take translation, for instance, a lot of things come together to work with the RNA polymerase. A lot of kinases come, they just add phosphates, they are gone. They just add some things and you don't see them again. Just for some short period, you don't see that interaction. It tells you that it will be hard to capture such an interaction. And so Co-IP cannot detect those weak or those transient interactions. Now, what if there was an indirect interaction? So protein A is interacting with protein X. Is there any cofactor? Is there any other molecule linking the two? We might not know. But Co-IP will just tell you that there is an interaction. So it doesn't tell you whether there was a direct interaction, like A is like with X, we can't say for Co-IP. So unless you perform downstream other applications, like in that case, you can perform GST-pull-down, which is one of the experiments that we can use to confirm whether there was a direct interaction or not. Now, the capture antibody for the bait protein may interfere with the bait-prey interaction. So here, like I was talking about the polyclonal and then the monoclonal thing, assuming the antibody you are using to target the bait, the epitope where this antibody is supposed to bind is the epitope that is, or is the site involved in the interaction with protein X. Then it tells you that when this antibody comes in there, it's going to disrupt the interaction between your bait and then the prey. And for that matter, you will not see any interaction between the two. So it's difficult when the antibody involved also recognize the binding site for your interacting protein. So that is one limitation. Now, whatever you do, you should also confirm or verify the interaction using other methods. So we have BioID, you can perform BioID Like I also mentioned, you can perform GST pull-down. There are a lot of other techniques that can be used to verify whether the interaction was true or not. And it's also qualitative. It just tells you that, yes, there is an interaction. You cannot tell the amount that is bound at a particular point in time. You can't tell. So that is one limitation. So now that is all about co-IP. We are using this paper as an example and how they utilize co-IP to answer their research question. So there's this protein which is RRP9. RRP9
Yes.
is someone
That,
okay?
yeah, I think before we move into the paper, there are some questions on the
Okay.
presentation first
All
that
right,
you
all right,
can
awesome.
just look at before we zoom into the paper.
Yeah,
The
yeah.
first
I
one
think
is,
you can
yeah, it
proceed.
said,
Whoever has a question can.
okay, so I think two are in the chat. The first
Okay,
one
let
is,
me see.
can co-IP be used to investigate other unknown molecules in a given sample? If yes, how
Unknown.
will the Western blot or gel be done since the interaction molecules are unknown? Yeah, so
Now, so if it's unknown,
mhm.
so if it's unknown, we don't have any idea. So we do basic how do you call it? Basic experiments. So let's say you are working with breast tissue and then you are seeing a new molecule. You have to do characterization. You have to characterize the protein that you've seen. Let's say two of them, you did live imaging and then you could see that they co-localize in a particular location within the tissue. You always see them in different tissues. You could see that these two molecules are there, but you don't know what these proteins are. You don't know them. What you do is that you characterize your protein. You can also sequence to know what the protein is made up of. Now, if we know the sequence, it tells you that we can develop antibodies against them. So that's the basic thing I think you should do. Now, if we're able to develop antibodies against them, now we can confirm whether they were interacting or not interacting. So that's one way you can do.
Yes, exactly. So thanks for the answer. Yeah. So this IP mean you have to know your two, your faith and your praise. Your
Yeah.
main research question is to look at the interaction. That's the main research question we are looking at when we are doing IP. To look at whether those two proteins that you are proposing or you've known, are they interacting with each other or not? So the kind of research question you are trying to answer will tell you which kind of research techniques to use. So thank you
Yeah. So
for that response. Yes, yeah.
if you don't know the molecules, you can't develop antibodies against them. So you
Yes,
should characterize them before you
yeah,
confirm whether there is an interaction between them or not. Yeah. The next
exactly.
question.
Yeah, as a follow-up to the above question, is there any lab in Ghana that is conversant with the use of SDS page? But I'm not sure. So
Okay.
maybe in the near future, our institute will develop lab in Ghana for you to be using. But myself, I don't know. But I'm sure maybe the KCR, do you do that, Daniel? KCR, do you do protein SDS page for protein, Angel?
Sorry, I cannot confirm that
Yeah,
for now.
yeah,
I think
yeah.
Evans is here. Maybe he can confirm that.
Is
Akwaba
Evans
Evans.
here? Where's Evans? Who is Evans?
Yeah. KCR do gel, but it's mostly for DNA stuff.
Oh,
Hardly
okay.
Okay, yeah.
protein.
So that is
Yeah.
just so there's difference between gel electrophoresis and SDS pitch. So I know even in our lab we used to do that's Sechi's lab at molecular medicine. We used to do gel electrophoresis. That's for
Yeah.
uh, that is for
We just do it for DNA
DNA.
and then RNA for the
Yeah,
nucleic acid.
DNA and RNA. That's gel electrophoresis. So.
Um,
But for Western blot, although we've talked about, but I'm not sure. I've not seen anyone doing that. I'm not sure about
so we
even
did.
whether Niguchi even do that.
So sorry, I didn't hear what Akwaba said. Evans.
Yeah.
Yeah, Akwaba said They only do the gel autophoresis. That is for
Yeah.
DNA and RNA.
Right.
But SDS page is also different.
So at the
Myself, I
Hive,
started doing SDS page when I moved here.
yeah, so the Hive, I don't know if you guys know of the Hive. The Hive, they do
Yeah.
SDS page. We did one with them some time ago. We had some
Okay.
workshop and then we did one. We expressed a protein for some enzymes, polymerase, yeah, DNA polymerase. We expressed it and then we used SDS page. Yeah. Even at SAS Lab, we do have all the equipment for SDS page, but we didn't get the chance to do it because there was no study that involved its application, and so we never used it. Yeah, yeah. But SDS page, I know Hive. If you have the chance to visit the Hive, yeah, I'm sure you should be able to see one and how it's done.
If not, if you get here, you learn. These are constant thing. Almost every week I do SDS page. Depending on kind of research you are doing, some of these things over the things you know for your life. So these are basic experimental things we do daily to prove that your protein is of interest is there. Yes, thank you for the question. Except there's anyone having any question again. Yeah. So I'm
Is
happy
there
a lot.
any
Yeah.
other
So
question?
I think I was having some questions.
Yes,
This
go ahead.
hour. I think, so I have a couple of them. Maybe if I think it's only left with 8 slides. So maybe if we can finish before I ask, or if you go on.
I'm sure
Okay.
you have other questions after this paper, so you can add it. You ask. No problem.
Okay. So I was thinking that how do we mimic the in vivo conditions for the interactions? Like
Okay.
maybe some proteins they will only interact in a physiological state. So you bringing them back into the tube. How are you going to know that in fact these ones are interacting?
Okay. So
Yeah.
what we do here is mostly we use mouse. So we have mice, and then you. So if it's breast cancer, we should generate a breast cancer in the mice, which is a phenotype of normal breast cancer, the breast cancer that we know. So let's say in this study, you suspect that protein A and B are interacting in, let's say, triple negative breast cancer. That's that's what you think. So you transfect these mice with triple negative breast cancer. Now, in some of the mice too, you are going to transfect them with the same triple negative breast cancer, but in a particular group, you are going to delete protein A, but with the other group, we don't delete protein A. Now we know that, let's say protein A and B, they are expressed in the liver. So we harvest the liver. Now we isolate the cells. So just know that it was expressed in a system, which was the mice. So if it's expressed in the system, we harvest the liver and then we perform all the analysis. Or we go through the whole steps as I mentioned. So no matter what, it will still come out of the system. Now, if you don't want to do it that way, we can also do immunofluorescence, whereby you take the cells. Let's say you cook water, your proteins together with the cells. Now, when the cells are growing, what you do is that you visualize, you stain, and then you visualize to see whether they are co-localizing. So sometimes in the nucleus, you could see that these particular two proteins, they always end up in the nucleus and they are kind of overlapping, or they are always together, whilst other proteins are somewhere else. So that's one way we can do it. So it's not just one. Over here, we don't do one experiment and then confirm something. We do series of experiments. We do the in vitro. You generate these conditions in mice to also confirm. You can also culture cells to also confirm. You can take normal human samples and then stain to see. You can do immunohistochemistry to see if these proteins are co-localizing. So we don't just do one thing. We do series of experiments to answer the same question. So that's how it's done.
Okay, thank you very much with that one.
Yeah.
So I think maybe you can do side and side, just as you are saying with the let's say fluorescence, and then they will show your
Exactly.
localization. Okay. So the second question has to do with the how do you take care of the post-translational modification of the protein? Let's say you see some proteins after they are being translated, a lot of things happen to them. Carbohydrates, other lipids are being added, a whole lot of stuff. So how do you get a clean form of the protein only, or maybe if it is in their modified state that you are hunting, like you are hunting for it in a modified state, or are you hunting for it in its pure state?
Yeah, so you see in the post-translational modification, one common thing that is being added is phosphate. And sometimes we have methyl groups being added. You can also have the acetylations as well. So we do make sure that we are aware that these modifications happens a lot. So mostly you confirm whether when they are in the unmodified form, is it when they are interacting, or when there is all the other factors like the modification with phosphate, the methyl groups, and what have you. So if we suspect that the modifications are there, then in our sample preparation, you make sure that you include like the phosphatase inhibitors. You know, phosphatase are ones that kind of dephosphorylate the proteins. Okay, you make sure you use inhibitors to mimic as it used to be in let's say in vivo, because maybe the interaction is only possible when the phosphate group is present. So you do make sure that, so all these things, before you even begin your experiment, you must know all these characteristics of the protein or the things you are interested in, else you miss it. Because if you miss anything,
so it can be that maybe if it has a multiple interaction, let's say methylation, the deletion just as this pi is coming to say, and then maybe phosphorylation, but you know that it's phosphorylation state that will help the binding. So that one you don't touch the.
You don't touch. You make sure it is
the
preserved.
phosphorylation, so no phosphatase inhibitors or something. Okay, sure.
So you have to maintain all those conditions, else you end up missing the interaction.
So how then do you come up with, let's say, a new idea that these two proteins are interacting, and then you are going to perform your experiment to prove the interaction, but you have to know the interaction first. So I don't get that one.
So basically, let's say I suspect two proteins to be interacting, and then, or let's say you were just staining some samples and you saw that there are these two proteins. When you perform your immunohistochemistry, they are always together. So that was immunohistochemistry. The tissue is now out. It has been processed. You've processed the tissue. Now you've stained and you are seeing all of them together. Now, if they are together, it raises some sort of suspicion. Why are they always together? You take liver, you processed, you did some sort of imaging. You know, sometimes you can culture cells, you did live cell imaging, and you are seeing all these things. Now, when you see them, that's your suspicion. So what I'm going to do is not just the interaction, but you add an extra question. So I suspect that they're interacting. That's the first suspicion. And you saw that maybe in a particular group, those with severe form of the condition, they are the ones where you are seeing the two proteins together. But when you have mild condition, the proteins are not together. And so that was what you saw at first. So you want to confirm. Being together does not mean that they are even interacting. I hope you understand. So you now want to go ahead and confirm your suspicion. Why are they always moving together? Why are they common in the severe form, not in the advanced form? Then you can say that, oh, okay, then let me see if they are interacting. Let me see if they contribute to the disease severity. Let me see when I delete them, what will happen to the severity? When I delete one, what will happen? Then you start asking all these questions. So it always starts from somewhere. Someone you come here, sometimes you come, they will tell you that there was this graduate student who was working on this project. These were the findings. What do you think? And the person had already made some kind of conclusions that A, B, and C are always working together. They are working together. Now you now ask the person, does it mean they are interacting? Does it mean they are contributing to the severity? Does it mean they delay the onset? Then you start to use these applications to confirm your suspicion.
Okay, that's very nice.
Yeah.
And then the last one.
Okay, so I think before that,
yeah,
another scenario I'll add to what he just said is it depends on where those proteins are. So, for example, you are studying lysosome and you find that the lysosome membrane has particular protein, let's say protein A. Then you find that there's also another protein that kind of has to be moved into the lysosome before it functions. So it produced somewhere, let's say in the endoplasmic reticulum, where most proteins are produced, so protein B. Then you find that, in certain conditions, those protein A, which is around the membrane, is there, and protein B, it kind of move into the lysosome. So maybe you want to see that whether protein B moving into the lysosome, it's kind of being trafficked or interacting with the protein A that's in the lysosome, so that they'll move into the lysosome to function. So the location of where the movement of those kind of different proteins will also help you to see that, this kind of interact with this protein so that they move together to a certain place to kind of work. So you have to kind of study to see the role or the location of different proteins, how they are working. Then you now process those hypotheses that, because this protein is moving from this way to here, and this kind of serving as a police, you want to see whether it's assisting this. Then you have to propose this IP as one of the research methods.
Okay. Thank you very much. That's nice.
Last thing, in the paper we are going to discuss, you will see that even though I did not show the picture, they were suspecting that one protein is interacting with a lot of proteins. So they did mass spectrophotometer. That's what they did. They saw a lot of proteins interacting with it. So they now went ahead to see if the RRP9 is part or not. Do you understand? Sometimes you can do mass spec. Now you see that, this particular protein, there are a lot of proteins interacting with it or around it at the same place where we are seeing it. It's also there. Is it interacting with the other ones? Then you do other things to confirm your suspicion.
Okay. Thank you very much.
You're welcome.
Yeah. So, and then last one, sorry, with the your protein A and X. After we've passed the gel through a membrane, we say that we would have to add an antibody that reacts specifically with protein X, so that we can confirm the presence of protein X.
Yeah.
So I was asking myself then, why the headache for you to go through all the process if you have an antibody that can react straight away with protein X and then you go away with it, but you have to pass through all the process before you come and capture protein X at the end.
So you see here, the focus is not to confirm whether protein X is present or not. The focus is protein X. Is it interacting with A? That is the question we want to answer. So now, when you go back to this slide, you could see that, let me use a pointer. I don't know if you can see my screen.
I can see your pointer.
Good. So here, this is protein X, this is protein A. We can just do antibody to detect X. We will say X is present, but it doesn't mean it is interacting with A.
Okay.
Do you understand? So if you want to find out if it's interacting with A, now develop an antibody against A. Come and wash. When we wash, if it is interacting with A, then it should that interaction should still be there.
Yeah.
So over here, we are seeing all these things here because of A, because we detected A. So whatever we stain for, when we see X here, it tells you that it came with what? A.
Okay.
It was able to end up here because it was part of A. Mind you, when we get here, we are going to wash. If you are not part of this complex, we are going to lose you. Okay.
Yeah.
So the thing is to confirm whether A is there, B is there, but are they working together?
Okay.
That's the question we wanted to answer.
Okay.
We can't just stain A, we can't just stain B differently and say they are interacting. No. It's in a whole cell, in the cell, we have a lot of proteins. It doesn't mean they are working together.
Sure. Thank you very much. I think we can continue.
All right, sure. All right, so that is that on co-immunoprecipitation. So this paper utilized co-immunoprecipitation to answer their question. So the RRP9, it is SNO RNA binding protein. So SNO RNA is one of the, types of RNA. It involves in chemical modification of RNA to form ribosomes. So when we are forming ribosome in the ribosome biogenesis, you require RRP9. It's part of the complex that helps to form the small ribosomal unit. Now, SMURF1 is also another protein. It is known to nedylate or ubiquitinate proteins. So when SMURF adds ubiquitin to a particular protein, you are tagging the protein for degradation. Now, when you add NED8 to a protein, so NEF can add NED8 to a protein. It can also add ubiquitin to a protein. Now, the outcome is that if SMURF1 adds a protein ubiquitin to you, you'll be degraded. You'll be tagged for proteasomal degradation. Now, if it adds NED8 to you, your activity is going to change. The way you behave in a cell is going to change. Now, all this activity is known for SMURF1. We know that it does ubiquitination. We know that it adds NED8, but we don't know whether it adds NED8 to this protein, whether they even interact. We don't know. So the study wanted to find out what proteins are interacting with RRP9. Is SMURF interacting with RRP9? And if it's interacting, is it adding ubiquitin or it's adding NED8? So that's what the paper wanted to find out. And if there is an interaction, if it's degrading it or if it's adding NED8, how does it function in cancer cells? Is it going to make the cancer cells resistant to therapy? Is it going to make them more aggressive? So that's what the paper wanted to do. So two different proteins, we wanted to know if they are interacting, and if they are interacting, what are the effects? Is it adding something to it? Is it degrading it? Is it making it more aggressive? Is it making it, is it changing its mode of activity? What is happening? So they asked a lot of questions. They wanted to know whether it's interaction, is it adding the NED8? What site, if it's adding NED8, what particular site on RRP9 is the NED8 being added? And even if it's ubiquitin, what particular site is it adding it? Because the protein is a whole, you can extend it, it's like a stretch, okay? So with various domains. So which exact location is it adding the ubiquitin or the NED8? So that's what the paper wanted to do. So first thing that they did, let me get this away from here. Good. So first thing that they did was to confirm if RRP9 and then SMURF1 are interacting. That is the first thing. And this result is coming from co-immunoprecipitation. So they use HCT116. This is a cell line. They express the proteins, and then they performed co-immunoprecipitation to confirm if the two of them are interacting. So like I mentioned, this is our input. The input is coming from when you prepared your lysate. The lysate that you have, you perform Western blot on it to see if the cells you have, the sample you have, Is it having protein, the RRP9 protein? Is it also having this? And yes, we can confirm that the two proteins we are talking about, they are present in the sample. Now, let's use any IgG antibody that does not recognize the SMURF and then see if there will be any band. Now, this is serving as a negative control. Ideally, there shouldn't be any band here. So you see over here, there's no band, but here we are seeing a small band. So in theory, we keep saying we don't see any band because it's a negative control. But when you end up in the lab, you see that it's something else. It's crazy. Sometimes you troubleshoot and you still see something, but you try your best to make sure that this band shouldn't be there. So this is something that shouldn't be there. But the paper, we can't just base on this and then disqualify the paper from publication. So that is one thing. But note that in a negative control, we don't expect to see any band as we are seeing here. So now what they did was they used an antibody against the SMURF1. So this is antibody against SMURF1. And then they performed the Western blot whereby they stained for RRP9. So you could see that after washing and then everything, when they stain for RRP9, we are seeing it here, which is a band here. So we see the RRP9. They also just wanted to confirm if SMURF was around. And so they did the Western blot for SMURF1. And now we are seeing SMURF1 over here. So antibodies for, I can take you back for you to appreciate it well. So let's assume that this is SMURF1, the red one. We stain for it, and because it is interacting with RRP9, after washing, we see SMURF1, we see RRP9, we perform Western blot. Because it ended up here, we are seeing RRP9. So that's why we are seeing this band. So this first one tells us that yes, there is an interaction between RRP9 and then SMURF1. Now, it didn't end there. They wanted to do the what we call reciprocal co-IP. Reciprocal co-IP in the sense that instead of using antibodies against SMURF to see if RRP9 is there, why not do it the other way around? Let's use antibodies against RRP9 and then see if SMURF will be there. So that's what they did here. They used antibodies. So if we are to go back to our workflow, you could see that here, instead of using antibodies against the red one, in the reciprocal one, they used antibodies against the yellow one, so that when we get here, we will use antibodies against the red one. So it's like the opposite way, just to be sure that, yeah, indeed, there is an interaction between the two. So that's what they did in the reciprocal. So this is what we call reciprocal co-IP. You use antibodies. In the first one, antibodies against this. We saw that this was present. Okay, let's turn it. Let's use antibodies against the RRP9 and see if we are going to see SMURF1. And now we are seeing SMURF1. So the two results confirms that there is an interaction between SMURF1 and then RRP9. There's an interaction. Now the question is, the interaction, is it direct interaction, or there is a mediator which is linking the two? And so to be sure that there is a direct interaction between the two, they performed what we call the GST-pull-down assay. So GST-pull-down assay, you just fuse your RRP9 with GST. So GST is glutathione synthetase transferase, I think, yeah. Now we fuse it with this. It has a very high affinity for glutathione beads. So you incubate it with the beads. Now what you do is that you also incubate it with the SMURF. And if there is an interaction, then SMURF should bind to this complex. So you see the complex and then you perform a Western blot to see. So here, this is our input. In the input, there is no RRP9, but SMURF is present. And so when we stain for SMURF, we should see SMURF here. Now, when you come to this side, RRP9 is present. That's why we are seeing the plus. SMURF1 is also present. And so when we stain, we should see SMURF1. We should also see RRP9. We should see it here because the two of them are present. Now, when we perform Western blot, so we immobilize this, we incubated with this. Now, when we pull down, okay, when we pull down, we want to see if the SMURF is present or not. So this is our result. You could see that when you don't have RRP9 in the pull down, SMURF will not bind to anything, and so there's no band for SMURF here. You know, usually SMURF, the his SMURF, this is the band for it. It's between 90 and then 70. But because we don't have this guy here, so this is kind of a negative control. We don't have our bait here. So we don't expect to see any band here. Now, when we have the GST-RRP9 present, we incubated, we perform the pull down, we are seeing a band for SMURF1, and then we are also seeing a band for RRP9, which tells you that, yes, the interaction we are seeing here is not mediated by anything. It is a direct interaction between the two. So GST and RRP9 and then SMURF1, they directly interact. So this study used Co-IP and then confirmed with GST pull-down assay that yes, there is an interaction between the two. It's not just an interaction which is mediated by something else, but it's more or less like a direct interaction. So that is what they saw. Now, the next thing, if there is an interaction, we know that when SMURF is around, it catalyzes ubiquitination, whereby the protein is degraded, or it adds NED8. So let's see, will there be any effect on the levels of RRP9? Now there's an interaction. When we delete SMURF1, what will happen to the levels of RRP9? Is it going to increase or is it going to decrease? So that's what they wanted to find out. So here, they wanted to know whether SMURF, its interaction with RRP9 leads to its degradation. That's what they wanted to know. So here too, they use CRISPR-Cas9. This is our negative control. So in our negative control, you stain to see if SMURF is present. So it is negative. So SMURF should be present. Nothing has been done to SMURF1. We have not deleted SMURF1. And so when we stain, it is present. Okay, so this is our negative control. Now in the positive one, that's what they did. The next one, they deleted SMURF1. They've deleted SMURF1. SMURF is not present. So when we stain, we shouldn't see it. But you cannot see that there's a small band here. Like I mentioned, in theory, we are not supposed to see anything. But many a times when you do these kind of experiments, you do see some of these things, which is very disheartening, but you still see it. So there shouldn't be any band here. And this is our negative control. SMURF is present. When they stain, you see that RRP9 is present. Now, when you delete it, RRP9 is still present. Nothing is happening to the levels. So over here, they could tell that when we delete SMURF, there's nothing happening to the levels. Now, the paper concludes that there's no increase in the levels because if SMURF is interacting with RRP9 by adding ubiquitin, then it should degrade it. If we delete SMURF, then the levels of RRP9 should increase, but here it's not increasing. And so SMURF does not affect the stability of RRP9. That's what they saw here. It is interacting, but it has no impact on its stability. Now, let's use tissues and see. So they use colon, they also use spleen from mice. Now, in the wild type means that it has SMURF. SMURF is present. Now, the KO is knockout. They've deleted SMURF. There's no SMURF. So the same thing, they did the same thing for the spleen. You can use, like I mentioned, CRISPR-Cas9 to delete the SMURF. You can use the 3-LOXP system in the mice. You can do that where it specifically deletes SMURF1 for you. So when they took the colon, those with SMURF1, we see the levels of, so this is SMURF1 is present. The levels of RRP9 did not change. When you even delete the SMURF1, the level is still the same. RRP9, the level did not change. When you take spleen tissues, okay, SMURF1 is present, RRP9 is present. When you delete SMURF1, it's still present, nothing is happening. So they performed an in vitro, they did in vivo, and they concluded that SMURF, even though it interacts with RRP9, there's no effect on the stability of RRP9. And that is one arm of SMURF1. It's whether it adds ubiquitin to degrade the protein, or it can add NET8. So now, what about NET8? Is it adding NET8? And so what they did was to also perform a Western blot. Now here, the same co-IP. Here, what they did was that this is our negative control. We shouldn't see anything here for the negative control. So we are not seeing any band here. Now, this is an inhibitor for NET8. So NET8 is added to a protein. There is this E3 ligase which add NET8 to the protein. So it is an enzyme. So we are using an inhibitor that will inhibit the addition of NET8. Even though SMURF1 catalyzes it, it needs this enzyme to do that. So they used an inhibitor. So here there's no inhibitor. Here there is no inhibitor, but we see a band. Okay, this is RRP9, but there's a smeared band alongside, which is high molecular weight. Okay, so this is our NET8. It tells you that SMURF is working, NET8 has been added to RRP9, and so we are seeing this smeared band, which is a very high molecular weight that has been added to RRP9. Now let's add an inhibitor, which will prevent the addition of NET8 to RRP9 and see if NET8 will be present. And so when they added this E3 ligase inhibitor for NET8, you could see that there was no band. So here they were able to confirm in vitro that yes, NET8 or SMURF adds NET8 to RRP9, and it is seen or shown as a smeared band on the Western blot. Okay, let's take a tissue and then see what happens. So they took colon tissue from mice. This is the wild type. Of course, this is IgG. IgG is serving as a negative control. There shouldn't be any band here. This is our wild type. SMURF is present. Now, if SMURF is present, they stain for NET8. Then we could see a smeared band on the Western blots. Now, when we knock out SMURF1, we don't see the band. So it tells you that SMURF1 is the one mediating the addition of NET8 onto RRP9. When you delete it, you don't see anything. When it is present, you see NET8 being added to RRP9. So that's how they were able to confirm that, yes, there was an interaction. The interaction does not affect the stability. It does not degrade RRP9. It rather changes its behavior by adding NET8 to it. When you delete SMURF1, you don't see any band. When it is present, okay, when you delete it, you don't see any band. When it is present, you see NET8 band that has been attached to RRP9. So the next question was to look at the exact location where NET8 is added onto RRP9. So this is the normal structure for RRP9. It has the N domain, the glutamine-rich domain. It has the WD4. So there are other aspects of the results that I did not show, but they did kind of a different test that showed that the addition was happening in this domain. So when you get the chance to read the paper, you could see that there's another, a lot of other figures that comes before this. And they were able to confirm that the addition happens along this domain. So what they wanted to do is, if the addition is along this domain, not here, not there, now let's find out the specific site where the addition is happening. So when you take this domain, on the N-terminal, it has about 1 to 7 lysine residues. So lysine, amino acid lysine, a lot of them here. It has about 29 residues. No, about 8 residues here. It also has about 13 residues over here, lysine. So whenever you see K, that is lysine. Now they want to see on position, let's say 166, 172, 180, which exact location is the NED8 being added? So how do we confirm this? Then let's take the lysine, mutate it into a different amino acid that does not support the addition of NED8 and see. So what happens is, when lysine is present, we are able to add phosphates, ubiquitin, and other things. Over here, when you want to confirm that, yes, this particular site is involved, let's say position 10, lysine on position 10, we can add a phosphate group to it. We want to be sure that it is true. And why not change the lysine to, let's say, arginine or to something else that does not support it? So when you change it and you don't see the addition of the phosphate, it tells you that, yes, that is the place that was involved. Now, when you change it and you still see the addition, it tells you that the addition is happening somewhere else and not the position 10. So that's what they did. They took position 1, mutated it to arginine, position 172, mutated it to arginine. So all these positions, they changed lysine to arginine because arginine does not support the addition of NED8. So when we mutate, so in one sample, we mutate only this. If we still see NED8, it means that it's not on this site. Now, when we change it and then we don't see NED8, then it tells you that that is the site that is involved in the addition of NED8. So they did the mutation, they had a lot of mutants, and then they performed the Western blots again. So here, NED8 is present in all of them. We have the RRP9. Here, this is just a negative control. There's no RRP9 here. There's no RRP9, so we shouldn't see any band here. There's no RRP9, we shouldn't see any band here. This is our wild type. Wild type in the sense that we still have the normal lysine residues on it. We have not changed any of the lysine residues. It's still lysine. So if it's still lysine, then we should see the band. So this is kind of a positive control. Now on position 166, we've mutated lysine to arginine, but we are still seeing the band. It tells you that this is not the site where nedylation is happening. It's a different site. Position 172, lysine is converted to arginine. We are still seeing a band. It's not this position that is required. Now on position 221, when we mutated lysine to arginine, we don't see any band again. There's no band. And it tells you that position 221 is crucial for addition of what NED8 onto RRP9. So that's how they were able to also tell that yes, there is an interaction. It adds NED8 to RRP9. Where exactly does it add NED8? It's on position 221, lysine on position 221. So that was how they were able to confirm that. So that's one basic thing. Over here in most of our exams, they will tell you that, oh, you suspect post-translational modification on this protein, let's say position 20. Design an experiment to show that, yes, that is where it occurs. Then just change the amino acid, which is lysine, change it to something else that does not support. And if you still see it, you tell us that yeah, even after mutating it to something else, you are still seeing it. And so that position is not the one involved in the post-translational modification. Now, if you mutate it and you don't see it, then it tells you that that position is crucial for the modification. So that's one key thing you can do. Just mutate it to any different amino acid and see if it supports it or not. So now we know that SMURF interacts, it catalyzes the addition of NET8 on RRP9. That is something that is known. What is its impact on tumor cells? When we have SMURF interacting with RRP9, when you have SMURF adding NET8 to RRP9, what happens? So here they took some tumor cells, they wanted to, they cultured the cells, then they look at the cell numbers, whether they are going to increase in numbers or they are going to reduce. So here they had a control whereby there is a negative control, which is our RRP9. So RRP9 is intact. RRP9 is present. Over here, they use the CRISPR-Cas9 to delete RRP9. And then they cultured the cells. You could see that when you have RRP9 present, you have a lot of cells growing in there. Large number of cells, a lot of them, which is here. So, but when we knock them out, few cells are here. It tells you that RRP9 kind of supports tumor growth, okay? Now, when you delete it, it inhibits the growth, and that's what we see here. What about cell invasion? That is the cells, the tumor cells, their ability to infiltrate other tissues. You see that when you delete RRP9, they don't have enough ability to infiltrate into other tissues or other tissues surrounding them. But when RRP9 is present, they have the ability to infiltrate other tissues. That is cell invasion. So when you have RRP9 present, it kind of supports tumor growth and also cell invasion. But when you delete it, it does not. So over here, when you find out that two proteins are interacting, we are looking at therapeutic aspect. What are the therapeutic impacts? What will happen when we find something to inhibit its activity? What will happen to tumor cells? If it is an infection, when we are able to target that particular protein, what will happen to the infection? Is it going to make it more severe? Is it going to reduce the severity? Is it going to cure it? So you don't just come and tell us there is an interaction and then leave it there. No, it has to be applicable to something. We want to see the therapeutic impact. So that's one thing. Now, what about cell or tumor volume? So they wanted to look at the tumor size, the volume. Then here too, you could see that when we delete RRP9, okay, the tumor volume decreases. But when you have RRP9 present, the tumor volume is high. And so they were able to find that there is an interaction, it adds NET8. When RRP9 is present, everything about it supports tumor growth, cell invasion, the cell volume, and everything. So that's how these people utilized co-immunoprecipitation to look at the interaction and also tell us that, yes, when it is present, this is its impact on tumor. They didn't end here. They went on to look at this in colorectal cancer, which I don't have the results here due to time. So if you get the chance, you can look for the paper online and then read. There is an update, the colorectal cancer part. It was published in 2021, but in 2022, they updated one of the images. I think figure 6, M and then N. They are saying that they messed up somewhere, and so they brought the original images. So when you search for the 2021, also look out for 2022, the updated images, so that you don't miss anything out. This is the paper, you can find it. And if you have any question, you can ask. Thank you.
Yes, yes. Thank you for the great presentation. And yes, myself, I've also learned a lot, and I'm sure everyone has also learned something. I think there's just one question in the chat. Please, with the stability, do you also mean it abundance?
No, so yeah, it's part of abundance, yeah. So degradation means proteases degrading the protein. So even when proteins, after translation, when proteins are misfolded, we don't want to have aggregates of them. And so they get ubiquitinated. Then proteasomes are able to degrade them. Sometimes too, you have these molecular chaperones like HSP 70 and 90 coming together to ensure the proper folding of the proteins. Now, if they are not well folded, we degrade. Degrade means like gone. So abundance, that is abundance, yeah. And then with the abundance, you can also look at the half-life, how long it sticks around. So it's abundance, but you can also look at how long it sticks around, yeah, over time. Yeah.
That's great, great. Yeah, any more question? If there's no question, I'll ask question for you guys. Okay, let's move to slide 28. I saw someone has a question. So let's go to slide 28. Okay. Yeah, so to make sure that you guys are also following, I'm going to ask you guys question. So in this slide, he said the mutation, they did the conversion of lysine to arginine.
Yeah.
Why do you think they changed lysine amino acid to arginine? Let's see. Anyone? Not any other amino acid. Because some also this presentation is one of the typical kind of styles we look at to when you move. Yeah, when you move here. So the interesting thing about the kind of research we do here, from the kind of research we do back in Africa, is that is the kind of data we generate. So here it's more of experimental. These are the kind of experiment we do here. But not that say back in Ghana, see, we almost analyze data and the rest. And the challenge we do, I say, oh, a different environment. I say, unya data, where you say, recruit to amount for once. Then when your data, and no. We come and do analysis, then you are done. No, you are doing experiments. So here you are doing a lot of experiments to make sure that you are seeing what you see. So if the experiment doesn't work out, Charlie, you are hot. So these are the kind of experiment we do when you move out from mostly the African geographical settings compared to the kind of research that we do back, which makes the work very interesting. So if you know the kind of research we do here in Ghana or Africa, anywhere in Africa, then you move here, since you're able to meet the two, then you are nice. Yes, Evans.
Yeah. So I think I will answer from the perspective of the amino acid being, I think all of them are positive and then basic as well, but per their
Exactly.
structures, I'm sure
Yeah,
they are
that's
mostly
the
found on surfaces.
exactly. That's just the basic
Yes.
answer. Yes. We want to still
Yeah.
maintain the function of it. So in this case, we are using amino acid that are within the same kind of, for example, the charges, they're all positive charges. So we don't want to destroy. It's kind of, we are still maintaining the structure, but the function is kind of a bit altered. The function and how they look like is altered. But the polarity is maintained in this kind of mutation. Yes, yes. So that's thank you, Evans. Yeah.
Sure.
Yeah, so any other questions?
Yeah, so to add, that is one thing we do here a lot in all of our exams. Design an experiment to demonstrate that this is it. So if you don't have the idea, if you don't know the properties of the amino acid, Charlie, you get everything wrong. That's one thing here. They want you to think. They want you to know if you want to mutate lysine to something, why not let's say serine, why not threonine, why not the other things? Like so you should understand it, and then when you come here, you won't suffer. Yeah. Because no one is going to ask you to list properties of amino acid. If you will teach you amino acid, but they won't ask you to group them into this polar. No, you are going to apply that knowledge. That's how it's done here.
Great. Any other
I think
question?
someone's hand was up. Is it Evans or is
Yes.
that
Yeah. Any other questions?
No, I think it was for the answer that I gave for the question that
Okay.
he asked.
Okay.
Yeah.
So yes, thank you very much for the great presentation. So I'm going to pause the recording here, and if there's another question. So this recording will be posted on our channel. Then you can always access it anytime.